Hi, everyone. Welcome to Quanterix's Fireside Chat. Thank you all for coming. If I could ask you to filter in a little, we'd like this to be like a fireside chat, or interactive and asking questions and in a kind of nice and relaxed atmosphere. So appreciate that. I'm Mark Roski. I run the collaboration effort at Quanterix, and I've been at Quanterix since near the start of the company. Many of you know me, and I'm really pleased to be here this evening with Professor Henrik Zetterberg, who kind of doesn't need an introduction, but I'll give a brief one anyway. He's the department head of psychiatry and neurochemistry at University of Gothenburg. And then he's also got many other appointments that it's hard to keep track of, although he's the professor of neurochemistry at University College London as well, where he runs the fluid biomarker group there. Henrik has over 1,000 publications in the neurodegeneration field. He's been responsible for a large variety of new assays. One of the things that impresses me most is that as we've been working with Henrik over the years, I've seen him launch the next generation of new neurodegeneration researchers, Alzheimer's, Parkinson's, et cetera, as those people have come through his tutelage and through his lab and gone off and established their own research groups. So that's really impressive, Henrik, and really appreciate you taking the time to have this discussion with us. So what we'd like to do is have a nice interactive discussion. I'm going to kick off with a few comments and questions to get the discussion going, but please feel free to pick Henrik's brain and let's all work together to see if we can use fluid biomarker advances to advance the field of dementia research in general. Our interactions at Quanterix started with Henrik about 10 years ago, and it was really an amazing situation where the small little company in Boston called Quanterix developed a new technology, Simoa, a single molecule array, and it enabled an ultrasensitive immunoassay. And we didn't really know what to do with ultrasensitive immunoassays. We developed 3 prototype assays, one for troponin for cardiac, one for PSA for oncology, and a tau assay. And we thought because that might be relevant for traumatic brain injury. So we developed these assays, and we could detect these things in blood and at lower levels than most other people, but we didn't know what to do with them. Through some collaborators, our head of assay development sent an email to Henrik and Henrik's colleague Kai Blenau, also a professor of clinical chemistry at Gothenburg, and said, hey, we have this assay, would you guys be interested? Here's the paper that talks about the technology. And Henrik and Kai wrote back and said, we never heard of what you're doing, and we're not so sure if it's going to work, but we're interested in trying. And then we said, do you have any samples that might be relevant? They said, oh boy, do we have samples? And at the time, we thought it was all about traumatic brain injury. We said, oh yeah, we have traumatic brain injury samples. We've got some samples from boxers. We've got some samples from divers. And we got all excited. This is super interesting. And then I think Henrik said, and by the way, we have some really nice Alzheimer's disease cohorts as well. So that got us off and running and working together collaboratively, where the group at Gothenburg has grown and built and used the Simoa technology as part of their key research tools, developed a number of assays that have been brought out into the field, and we continue to collaborate. We're very excited we've just recently launched a brain-derived tau assay on Simoa through a collaboration with Henrik and his team in Gothenburg, and we continue to work on next-generation assays. So we're really excited about that, the future, and to kind of kick off the questioning, maybe I'll focus a little bit on tau, just because that was the first one we worked on. So,
but could I comment on what you just said?
Please, absolutely, that's the point.
My recollection of this is it's exactly like you said. But the thing was that this email that came, many of you get emails about novel technologies and so, but this was a special email. So it was sent by Dave Wilson. It was relatively short. It described a technology that he thought we would be interested in, and he described something he called a compartmentalized detection reaction. And when I read about that, sometimes it's so hard to understand how an assay gets sensitive. But when I read about these beads and the microbells, and how one could pull one bead into one microbell and measure fluorescence from individual beads and see some beads lighting up and most of them being not emitting light. Then I was thinking that this is understandable. I sort of get it. And that doesn't happen often. That sounded bad. But and then the company was based in Cambridge and Boston, next to the Cambridge Brewing Company, making beer. So we were invited over. I went, and we saw the naked prototype instruments. I got to know the technology even better, and then we discussed science in this brewing company. So I think that those two things
I
were...
missed those facts. Those were pretty important facts.
Exactly. And we didn't believe that this would work as well as it did. So what we said, okay, let's test something where we know there is massive brain injury. And if these guys can't detect it, then we extinct it.
Yeah, right.
So we had a collaboration with researchers in Uppsala who were interested in hypoxic brain injury after cardiac arrest. And that's a major, major clinical problem. And cardiac arrest is not uncommon, and the mortality rate is 90%. And those who survive often sustain very severe brain injury. Then a researcher called Sten Rubertsson in Uppsala was doing systematic studies on people who had out-of-hospital cardiac arrest, were resuscitated on the way into the hospital, and came to the emergency unit with circulation but unconscious. And then he systematically followed these patients with repeated blood samples, a lot of EEG measurements, clinical testing, and then he could group the people into good and bad outcome. And this total tau assay that you had developed distinguished these people with almost complete certainty at admission. So it was, at admission one could get a hint, but the best thing was to wait 24 hours and look at the change in the biomarker level. And this was extremely interesting. And you also had a prototype, A-beta.
Right,
assay,
we did.
Both A-beta 40 and 42.
40, 42.
And this also became, rapidly we could see that this is actually working.
Excellent. And so we've come some way from total tau to now, as you can see in this meeting today, everyone's talking about p-tau 217.
Yeah.
And so what do you see? What in the meeting today and the work that you've been doing, what do you see as kind of the additive assays now beyond just tau? Well, I shouldn't say just beyond just tau, because I want to talk a little bit more about tau.
I mean, tau is such a good example, because tau, the assays we have had access to in cerebrospinal fluid have been total tau and one phosphoform of tau. And they work really well to detect Alzheimer's pathology. And you also know that the CSF beta 42/40 ratio is now really an approved test. And these measurements can be done with a lot of different technologies. But then tau is, beta amyloid is a relatively simple molecule. We heard yesterday that someone called it the peptide from hell, beta 42, because it's sticky and so. But it's actually a relatively, we know exactly what to measure. It's a 42 amino acid long protein with no modifications, basically. And so from that standpoint, it's pretty easy. And then, but with tau, with all the isoforms, it's a much larger proteins, all the different phosphorylation sites. There is also glycosylation, there are truncations happening. We have, when starting out with the total tau assays, it became quite clear that if you measure very specific forms, sort of subforms of tau, then you could get information beyond what was possible with the total tau assays, and also with the most standard phospho-tau assay. When you measure these subforms, you automatically lose a lot of molecules, of course, so the assays need to be very sensitive. And I think that is why you played such an important role in the early blood tau work, right, because it was not possible to do these measurements without the technologies. So the proof of concept studies and developing tests on Simoa for the different subforms, this opened up the field. And then there was one more thing. since we run a laboratory where we are making assays ourselves, having access to the possibility of, and this might come from the brewing company in Cambridge, home brewing,
Yes.
has been instrumental. And to have an instrument where you can play around yourself together with team members and tweak and test and develop assays, validate assays, polish the assays, that is enormously powerful. And a very important contribution to the field.
Excellent. And any more specifics about other tau isoforms that would be interesting and additive to the work we've done around p-tau217?
What has become clear is that these different phospho-tau forms, they carry some information which distinguishes different stages of Alzheimer's disease. And you all know now that, I mean, it really looks like the new treatments against amyloid should be initiated at the stage where the tau pathology is not too severe. And it looks like 231 to 17, the phospho-tau forms, that they change a little bit differently during the disease process. And that the level of change, the magnitude of change also gives information on how advanced the disease is in the brain tissue. And there are research groups now who are trying to use these different phospho-tau forms to stage Alzheimer's disease from a biofluid. This has traditionally been done with tau PET, and I really think that is important. And we should continue to work with both tau PET and correlating the plasma biomarkers to that. So that will be important continued work. There are also some data indicating that one, through the absolute concentration of phospho-tau217, could almost make a tau clock, describing how long a person with a certain level have had tau pathology for, and that can also be transferred into an amyloid clock. The data are a little bit less solid at the moment.
That's really
compared with amyloid PET and tau PET, but this is happening. And now it turns out then that tau phosphorylated amino acid 205 might have a stronger relationship with tau than 217 and 181 and 231, which really look like markers that change first and foremost in response to amyloid pathology. And what we have learned through the assays that exist on Simoa now is that when amyloid pathology develops, you almost immediately get a neuronal reaction to that pathology, which involves tau phosphorylation. And I have sort of started to think about this as a potentially mechanistically related to how neurons get ill from amyloid. So amyloid might give a pruning signal to synapses, and then you get phosphorylation of tau, so the tau lets go of the microtubuli. This axon can retract, and the neuron will be deprived of its axon and its synapse, but still alive, and this neuron will release the phosphorylated tau, the N-terminal part. And the C-terminal part might go to the lysosome or to the proteasome and get degraded. And when this is overrun, you have free-floating MTBR containing tau that can form tangles. And then the neurons die. And then they release total tau, perhaps.
Right.
Or brain-derived tau. And the difference between brain-derived tau and total tau is that when we measure in blood, we have a problem that we didn't know about. That I didn't know about. Others knew about it. Big tau. The tau form that has an extra domain encoded by exon 4A, which is between exon 4 and 5. And this big tau is approximately 200 amino acids longer. And tau experts have known about this for a long time. This tau is expressed a lot in peripheral nerves. It's in the enteric nervous system. It also might have something to do with ALS and motor neurons sending out axons in the periphery. And that is why the total tau assay worked beautifully to detect acute injury, but it had an association with neurodegeneration in Alzheimer's disease, but not at all as good as total tau in CSF. But then eventually, we and also Indianapolis researchers came up with this idea of making an antibody specific to the junction site between exon 4 and 5 encoded sequences. And that is what became brain-derived tau. And then this is not, it can't measure big tau. So that sort of, I shouldn't say interference, because it's not really interference, but that confounder is gone now. Yeah,
Please.
I have a question.
yeah.
I've already asked it once today.
Yeah, we heard you.
Here's Mike.
You just explained that the peripheral sources of tau. You've just explained that the peripheral sources of tau are contributing to the amount of tau that you find in the brain that doesn't have any phosphorylation on it.
No.
And yet, what they're claiming is that if you measure phosphotau 217 and you divide it by the same peptide derived from blood-derived total tau, that improves the precision of your measurement.
Yeah.
But I would think that because it's being contributed to by peripheral tau that has nothing to do with the brain and doesn't correlate with CSF tau, doesn't correlate with A beta PET, doesn't correlate with tau PET, that would increase your variance and not reduce your variance. So can you explain to me why that works?
Yeah, but we don't like the ratio for that specific reason. So we love concentrations of individual proteins. And then you also in clinical chemistry practice, I mean, we run a clinical laboratory in Gothenburg. It's much nicer to work with individual concentrations and measurements and look how that relate to each other before making a ratio. But I must say that the exception is, of course, A beta 42.
If you measured brain-derived tau, however, I think that might actually do it.
Yeah, but still, it's a little bit dangerous because you might have seen the recent publications on big tau also becoming apparent in the CNS, and it looks like there could be interesting differences with ALS. So I think what we have done now is that we have made a blood test that gives the same information as the old CSF total tau test. And that will most likely be useful, I think.
I have a question too. So first of all, thanks for having this gathering over here and allowing us to ask questions and learn. I have a disclaimer, I'm not a neuroscientist, I'm an immunologist. So
We were going to get to that topic, so good.
Okay, 2 questions. The first one is, I'm old enough to remember when we had only CD45, a leukocyte marker. We didn't know what to do with this. And then when we started to be able to have technologies to look at isoforms, mainly CD45, RA, RB, RCRO, and combination thereof on the same cell, we started to understand the biology.
Yeah.
Effector cells, memory cells, migrating to this organ, that organ. So my question is, when I learn about p-tau and all these isoforms, we're very good. We have different platforms, antibodies to look at phosphotau on different epitopes, whether it's 217, 181, 231, etc. But I have never seen someone looking at, let's say, phosphorylation pattern on a single molecule to understand not at the population level, which is one thing, but at the single molecule level, which could be a lot more predictive than looking at population. So that's my first question. Second, depending, Henrik, how are you going to be answering the biology? Would Quanterix technology be able to, A, pick up with, let's say, molecular barcode at the molecular level, and second, could you actually multiplex to identify those variants?
Yeah. Yeah, the first question is a wonderful question. So it's, and I think this is the thing with having I mean, we talk about Alzheimer's disease as having hyperphosphorylated tau. The traditional assays we have been using they measure phosphorylated tau. But for example, diphosphorylated tau is possible to measure, but it's, for one reason or another, it's hard to get the sensitivity right. But we see it in brain tissue, multiply phosphorylated fragments where it sort of insists, it's on the same molecule. And those are they are only seen in Alzheimer's. They are not, the phosphorylation of tau, that's something we all do. And that's, I mean, the highest phosphorylation levels we can measure are in newborns who are turning over their synapses a lot when during the last trimester and the first year of life. So that's sort of also a physiological mechanism. But these di- and triple phosphorylated tau molecules, they are really, it seems like they are really pathological. And, but in the biofluids, we can't really measure them in a reliable manner yet. But I don't think we should give up. I think we should continue to try to measure it. Is there, Laia, would you like to comment from your mass spec perspective?
I think we've tried with the CSF, with the...
You might need a mic.
Yeah.
So as Henrik explained, someone in the lab measuring brain and D-phosphos and that those can be measured. But in CSF we tried, but it's just like the concentration is so low that it's very, very hard. But we are working on a targeted method to measure these D-phosphos in CSF, not yet in plasma. And also you have a publication from the group from Juan Lantero Rodriguez. He described like 231 and 235. So 235 can only be phosphorylated if 231 is phosphorylated first. So you can check out that publication. So there's like some logic behind like accumulating phosphorylations in a single peptide or very close to each other. But at the molecule level, I don't know if Henrik or someone from Context, how could we isolate one single protein and measure that?
There is actually another assay on the Simoa platform that measures 217, where the assay also has some reactivity to, it gets even better if there is diphosphorylation on 212 also.
Yeah,
Yep.
but then you're getting into KD and not
Exactly.
Exactly.
That's tougher. Yeah, I agree. But still, it's.
But you kind of brought up the inflammation link too, and I wanted to kind of drive that.
I can just mention
multiplex.
also that this idea with combining a lot of these different well-validated single-plex assay onto a multiplex.
Yeah,
that
exactly.
is, and I think you are thinking about making a tau
absolutely.
toolbox assay, sort of.
Absolutely, we're definitely doing that. And we can currently multiplex on the existing technology at about a level of 4-fold, which isn't probably enough, but we have a next generation platform that's going to go 15, 20-fold with the same lossless resolution so that we should be able to, and that platform is going to have more sensitivity than the current platform. So that should get us closer to this idea.
And then one could think that this will be extremely difficult to evaluate. But we have done one experiment in the Gothenburg group, and that is to simply dichotomize to a cut point and then count the number of positive phosphate sites. And that correlates surprisingly well with tau PET stage, which is pretty amazing. So it's this, what do you call it, this disturbance plot. It's relatively easy to make a little bit of a user-friendly readout on how progressed the patient is. And then it could be that it's the first 2 stages or so who would be, that those patients, that there you have the sweet spot for treating.
Well, that was great discussion around tau, and maybe I just expand a little bit.
Can we continue on tau?
Sure.
A little bit more. Because it's the, now you got to meet.
I was trying to switch you, but I'm perfectly happy to.
I would like to say one more thing, if you're fine with that. There is such a difference between N-terminal tau and the microtubule binding domain and C-terminal tau. And that we've also learned through these assays. At least we learned, but others might have known it from other ways. But anyhow, it's really, there are now indications that if you, instead of measuring the N-terminal tau, which is released actively from neurons that are ill from Alzheimer's disease in an amyloid-related manner, if you also measure more C-terminal fragments, there are some data indicating that you could get an A-beta 42 to 40-like effect with a selective loss of the more C-terminal forms. If you divide it by, now Dave Morgan will remind me that I didn't like ratios, but that ratio is then lower if there is tangle pathology. Sorry, I really, you got me there, Dave. Yeah, exactly. 42 to 40, I love 42 to 40. In CSF, but not in blood.
So you're not a ratio bigot.
Exactly. No, that's right. And then there is another tau form, which has attracted a lot of attention recently, and that is through Randy Bateman's team, this tau MTBR243 molecule. And when they explain what they have found, they describe it like they have found a tau fragment which is released from tangles through some type of enzymatic cleavage. And then this fragment is positively correlated with the amount of tangle pathology by PET. But the details, I don't dare to discuss that. It's probably Rand and his team who need to figure that out. But that's another way of moving a little bit more C-terminal to the tau molecule, giving you information, not just on this amyloid-driven release of phosphorylated tau, but also on the growth of tangle pathology. And potentially that could be useful across tauopathies, because, I mean, Alzheimer's is then tauopathy secondary to amyloid, but then we have a host of really important diseases that are seen in the memory clinics, which are then primary tauopathies, but tau is the pathological hallmark related to neurodegeneration, also in the absence of amyloid. Now I'm done.
Now
on tau. Now I will.
I guess I'll just ask one more thing related to tau to keep the tau thing going now. You know, relative to the combination therapy focus of this meeting, I was just thinking the tau story might be really useful there as you try to develop biomarkers. The situation is more complex with combination therapies. Do you have a view on?
Yeah, I mean, combination therapies could look, so there are so many potential combination therapies. And of course, people are looking at uncoupling tauopathy from amyloid pathology. Because if you remember this fascinating case from the Colombian kindred, a mutation, presenilin mutation carrier who lived up to her, she was 70, 75, and had amyloid pathology, but very little tau pathology, neurodegeneration. And she was then found to have this ApoE Christchurch mutation, which gave a little bit of a hint on molecules that could mediate amyloid effects on neurons. So you could sort of be a person resisting tau pathology. And then reelin is another molecule that has appeared in this context too. So I think that could be, when we think about combination therapies, there could be, there are many research groups looking into if one can inhibit whatever amyloid is doing to the synapse, potentially together with complement and microglia and astrocytes, and not make tau get phosphorylated and let go of the microtubuli. And then one would have a brain where the neurons are not retracting their axons and synapses, even if there is amyloid. So there we could think about kinase inhibitors and a lot of different things where specific tau forms could be really relevant. And it's been very interesting also to see, Down syndrome, that you have this triplication of chromosome 21, where DYRK1A sits, and DYRK1A phosphorylates at amino acid 212. So you see here how interesting it is to have different
tools in that toolbox.
Distinct tools, and dissect these different things with high precision.
Henrik, you mentioned something about the primary tauopathies.
Yeah.
Do you have anything to say about new biomarkers that might distinguish the primary tauopathies, or conversely, a biomarker for discriminating CAA?
Yeah, exactly. So the primary tauopathies, one good thing is, or not good for the patients, but they go with increased plasma neurofilament light quite distinctly. Tau positive F3, progressive supranuclear palsy, CBD, you capture the neurodegeneration with neurofilament light. But the tau tests are currently lacking or lagging behind. But there is one observation that I know several of you have seen it also, that the Alzheimer-related tau biomarkers, they look a little bit depressed in the biofluids, but it's not diagnostically useful. But there is something, a little bit, they look almost more normal than controls, but not, of course, that's not normal then. What has emerged, thanks to wonderful work by Anja Schneider's team in Bonn, she has revisited the blood vesicles. That has been a very difficult field with a lot of contradictory results. But according to her protocol, if she takes and isolates vesicles from patients with primary tauopathies. And if she lyses those vesicles, then she gets, and measures with a total tau, so it looks, I think, then she gets an increased concentration, which looks really promising. So I think that could be one way forward, and also, of course, looking at the more C-terminal parts of tau, and see if one could get something good coming from that. And I'm very interested. I don't know if it has been done yet, but this 243 fragment, I don't know if that actually signals from any angle, or if it's just Alzheimer's. I have not heard about that yet or seen any presentations. But those markers will be important to explore in primary tauopathies.
How about other ARIA-related biomarkers?
Yes, yeah, that was also a question. So this is a burning issue, of course. So either predictive biomarkers of ARIA or biomarkers that can detect dangerous ARIA. Both of those will be extremely important biomarker development projects. And there are interesting signals in cerebral amyloid angiopathy. If one measures the plasma A-beta peptides, A-beta 38, 40, and 42, it looks like you have lower levels of all of them in plasma if you have a lot of amyloid in the vasculature. But no one has, and if you look on all the A-beta 42, 40 data that exist from many, many different cohorts now, many of us have been focused on the ratio between 42 and 40 in plasma as well. But if you look at the raw concentrations, you will see that there is a tail of people with abnormally low A-beta 38, 40, and 42 concentrations. And I think it's a reasonable hypothesis to test that these might be the ones who have amyloid in their vessels. But it has not been proven, so it's really just a hypothesis. So if you sit on data like this, look carefully for that I would say. And I also try to say to all the companies that if they could look at the baseline A-beta 40 and 42 concentrations and then take out these people and look at the incidence of ARIA and see if that goes with an increased incidence of ARIA, that would be also extremely interesting. This idea with dangerous versus not dangerous ARIA, I have wanted to use NFL and GFAP and see what that looks like. But I have never seen any data. And but I can't think that ARIA that really affects the neurons wouldn't go with an NFL increase.
You would think, yeah.
It should do that. But I'm not sure. I haven't seen any data. I mean, they work in stroke, they work in subarachnoidal hemorrhage, and if we then think about these terrible few cases with macro bleeds and so, but that's too late, of course. You see it on imaging. Right. You have to have something predicting it. So I think we need to look at genetics of ARIA and fluid-based biomarkers for risk of ARIA. That is cerebrovascular disease, I think, and then onset of dangerous ARIA, hopefully something that, so that one could use these markers much like it's done with natalizumab in multiple sclerosis, where you actually can use plasma NFL to predict onset of a side effect of the drug.
I knew he was wanting to ask you another question.
So I've got a little question. You know, you're talking about ARIA and the seriousness of ARIA, and you know, we do this all the time with head injury now, you know, and CSF. But I'm not aware of any work that's looked at CSF markers of like albumin, fibronectin, any of the things that you would look at to see if you've got a major leakage or a minor leakage. Are you aware of anything that's done that yet?
No, but I mean, these are two good assays that do exist. So looking at the CSF to serum albumin ratio would be an almost obvious thing to do. And see, again, I think one could stratify the patients and look for differential incidence of dangerous ARIA.
But you'd have to do it in Europe because Americans won't let them do spinal tap.
No, exactly. No. So but in Europe we don't have the drugs. It's so sad. But I must say, I think this is, this thing with having access to drugs and learn about the drugs in a careful way together with the patients and clinical colleagues, it's extremely important. And I think we should try to figure this out to investigate and initiate trials if the companies do not want to do it, to find out the predictive and diagnostic markers of dangerous area. There are several other candidate proteins that need to be explored. And of course, it's the placental growth factor that was discovered in the Mark VCID project that Donna Wilcock and some other researchers have done really great work. And there is a CMO assay for placental growth factor.
There is, yeah.
And then we have, of course, the astrocytic end feet on the neurovascular unit with GFAP. But GFAP will not be, it feels like there should be a GFAP abnormality in ARIA. And then we have aquaporins, a lot of proteins that are enriched in the blood-brain barrier that we should look at in this context as well. So I think if I would like to embark on a PhD project or so, to have this concept of finding or improving or developing biomarkers for cerebrovascular disease, CAA, and ARIA, that would be such an important project.
Excellent. Well, maybe if we could just kind of wrap it up by, maybe you could comment. I know your team has been working hard on, as these key tau and other fluid biomarkers are moving towards the clinic, there's some implementation issues.
Yep.
So you had some excellent presentations earlier today. Could you talk about what you're doing in that regard about implementing fluid biomarker assays more conveniently, I would say?
Yeah. So I think almost everyone agrees now that plasma phospho-tau217 holds clinically relevant information. So it's really, I think it's just a matter of time until we will see these assays up and running in clinical laboratory practice around the globe. So it has such a strong signal. But then there are things that are underexplored and that are a little bit disturbing that we need to figure out. For example, it looks like some ethnicities are associated with lower increase of phospho-tau217 and other phosphotau markers when you stratify according to the amount of amyloid pathology. And that could be related to different ApoE haplotypes. And this we need to figure out. And it might be that one needs to develop other markers to compensate for that. There are discussions on kidney disease and active cardiovascular disease, or decompensated cardiovascular disease. These bring up the phospho-tau levels a little bit, but they don't destroy the diagnostic information. But if you're a gray zone individual, you might, of course, cross the threshold. Many people are thinking about implementing a two-cut point approach, leaving a gray zone where we simply will say that we can't tell if it's pathological or not. But this needs to be also set in stone. Not set in stone, it should be re-examined, of course, but we need to figure it out and keep an open mind. And then we also need to find, there are a couple of things that I feel are difficult as a clinical chemist also. There are some people who have high phospho-tau levels in blood without having amyloid. And we have even seen in a study on relatively young people, I think there were 78 or 80 young people who gave blood samples every week. Then there are 2 of these people who spike twice during that time for both phospho-tau181 and 217. And it's not a little spike, they spike into the pathological range and then they go back again. And why that happens, we don't know. But we hear this from our colleagues also, that there are occasional flyers. And it's not analytical, it's something biological that we don't understand completely. And then we have another very fascinating finding, which we discussed perhaps a little bit too little. In ALS, you have increased phospho-tau181 and 231 in plasma, but not in CSF, which could be super useful if this reflects some type of motor neuron degeneration related. Perhaps this is something one should use as a biomarker in ALS. But it could be, of course, a confounder if one is not. It really shows that you really have to have your clinical mindset and make sure that you are interpreting the biomarkers in a correct context. 217 seems to be less vulnerable to this ALS effect, but the ALS effect could also be something positive. Clinical implementation. We also need to try to standardize these assays to make them more comparable. And now Alzheimer's Association and ADDF are actually just about to fund an IFCC, International Federation of Clinical Chemistry and Laboratory Medicine, standardization project for phospho-tau 217.
Excellent. Yeah, we were very interested in that and getting the right standards and making sure the assays are manufacturable and reproducible.
Yeah, exactly. It's, should we talk about inflammation?
Yeah, well, I think we've gone a long way. And I appreciate all the discussion. And it might be just excellent now to have a beer and interact with people. So we take you off the hot spotlights, Henrik. But we really appreciate our collaboration. It's generated a lot of great research and moved the field forward, and we'd like to continue working with you. And thanks for coming. And
and thank you for doing this and for keeping the homebrew assays going, because
Excellent.
that is that's amazing.
excellent. Thanks. Thanks, everyone.